So, good afternoon and welcome everybody to this first of the international webinar series that is organized by ICBAS. Other webinars will follow. Please be aware. I think we will start in the best possible way with our guest of today, Professor África González. Welcome to ICBAS, place that you know well. And ICBAS thanks you very much for the support that you have given us in several occasions. Professor África González is a medical doctor. She graduated in the University of Alcalá de Henares back in '86, and she defended there also her PhD thesis in '91. And Professor África completed her residency training in immunology in '91 at Puerta de Hierro Hospital in Madrid. África then moved to Cambridge, and she had the opportunity to work and the privilege to work with in the laboratory of César Milstein. For those of you that are not as familiar with immunology, César Milstein received the Nobel Price for the co-development of the technique that allow obtaining monoclonal antibodies that are now widely used for different medical applications and for lots of other applications as well. Everyone that works in science, in the health sciences, knows how useful and powerful monoclonal antibodies are. Professor África González then returned to Spain in '96, where she started the area of immunology, actually in Galicia, and particularly where she is now located at University of Vigo. There, she directed for 10 years, up to 2019, the Biomedical Research Centre, CINBIO, at the University of Vigo. And actually, Professor África was the first director of this institute. Currently, Professor África González is a full professor of immunology at the University of Vigo. And apart from the more academic activities, Professor África is also a recognized entrepreneur, as she co-funded the spin-off Nano Immunotech, a company that works with nanosystems for creating biosensors. And this particular work of Professor África led her to the final stage of the EU Prize for Women Innovators 2017. So Professor África went to Brussels for the award session, if I remember well. Professor África is well recognized among the scientific community in Spain. She presided the Spanish Society for Immunology from 2016 up to 2020. And África has a vast published CV that recently included the book that you see behind África, that I have here as well. It's a very, very nice book that can introduce you to the area of immunology. And África is not only an excellent writing science communicator, but also a very nice speaker, as you will see in a moment. So, África, thank you very much for having accepted starting this international webinar series at ICBAS, and we are anxious to hear from you. So,
Thank you very much, Manuel, for these nice presentations. We are friends from many years now, and for me it's a pleasure. It's a great pleasure to be the first to start these international webinars that I'm sure that they will be a success. And also thank for the introduction of the book. It came this month, so it's just very recent, and I'm really quite happy with the results. So I hope that people would like to read it. When you invited me, you told me talk about whatever you want, and I decided to call immunopower because we have been in this pandemic, and when I was president of the Spanish Society of Immunology, that I was just nearly finishing is when this pandemic started, and it was really a revolution, and it's also now our role trying to answer questions for the journalists and trying to understand what's going on, especially with the vaccines and with the how the immune system is working. So I decided to talk about the immune system in general, but in relation with the pandemic. So, well, I hope that you would like the talk. Well, first of all, in 31st of December, Wuhan talked that there was several cases of pneumonia, and we were just astonished that in 10 days they were building a new hospital. And I remember that in Spain, we were just thinking, well, Chinese people are able to make this kind of huge buildings, and maybe in Spain could be completely impossible. Just in the 12th January of 2020, we have the sequence of the virus. And just on 11th of March of 2020, the president of the World Health Organization decided that this was extended along the globe. This was a pandemic. So we should not forget that this is coming from a zoonotic. There are viruses that affect especially bats, and bats has been the original of several zoonoses like rabies, Nipah, Ebola, and coronavirus. But coronavirus, there are different types. We have several ones that are producing common cold, that every 3-4 years we are suffering from some of them. However, very recently in this century, we have been infected by 3 different coronavirus, one in 2003, and 2012, and the SARS-CoV-2 in 2019, producing really severe respiratory disease, and this is the reason that is called SARS-CoV-2, producing really an important problem, especially in the lungs. Well, the virus, it's quite well known, has an envelope, and is one of the viruses with a biggest RNA genome. And this is important because in the replication is maybe it can include errors, but somehow they are able to repair part of them. And in the envelope there are one protein that is prominent, is the spike protein that many people have been listening about because the vaccines has been developed thanks using this spike. Originally, it was described that the virus needs the ACE2 receptor to go inside the cell. However, we have some proteases helping the virus to go inside. This seems something very strange, that we have elements helping the virus to be internalized. But we have several, and the first one that was recognized was this element, TMPRSS2. However, more recently, it was seen that furin and cathepsin are also playing a role. So the virus internalize, they release the RNA, and these are able to produce more proteins and then to release more virons. As I said, the virus has a long RNA, but is able to correct the errors. And in fact, this virus has lower mutations than HIV or flu, and the number of mutations per sample worldwide is not very large. Now there is a consortium, so several sequences have been included, and this has been able to check how different viruses have been expanded along different countries. And we have to say that the original Chinese variant has changed very quickly, because the original SARS-CoV-2 in the position 614 changed in this February and started to be expanded along different countries. And this variant is much more infectious than the original one that appeared in China. So this has been called the European variant. Now they have described thousands of variants, but not enough to be considered new strains. However, for example, in flu, the change of the virus is so high that we are having different strains every year, and that's the reason that we have to change the vaccine for flu. The World Health Organization have now defined the variants with Greek letters, just not to have the connotation of the countries that originally were called. So now we have some variants of concern, alpha, beta, gamma, delta, and some of them are of interest, so we should follow them. But at the moment, they have not shown that it could create a very high health problem. So we can say that against COVID-19, our immune system works. Why we can say that? Well, because there are many people that can become infected, but many of them are asymptomatic. And from the symptomatic, 80% do not develop severe illness. 15% could be in severe and 5% could be critical, but just from the symptomatic people. So our immune system works. And what is what our immune system is doing? Many people think that the immune system is a defense mechanism against pathogens, but the immune system is much more, it's many more. It's able to discriminate between foreign tissues, so it's doing the non-self differentiation, and is able to recognize tissues or organs in incompatible transplantation. Also recognize endogenous danger signals. For example, if we have an accumulation of uric acid, we can have inflammation because they are recognized by danger, like endogenous danger signals. Another thing that the immune system is doing is the antitumoral immunosurveillance, is doing a vigilance of the tumoral cells, and they are able to recognize them and to destroy them. And also something that we forget is tissue healing. Of course, there are main factors affecting the immune response, and there are some that are related with intrinsic factors that we cannot change. For example, our age, there are immunosenescence, the genetics that we are having, the sex, comorbidities, previous exposure to other coronavirus or to a vaccine. There are factors related with the virus itself, the amount of virus that enter into my body, the viral charge, the virulence, the variant, and also the evasion mechanism that the virus can have. And there are also factors, extrinsic factors, for example, drugs that we are receiving for the different illnesses, the nutrition. Malnutrition is considered the most common immunodeficiency, secondary immunodeficiency, because the immune system needs a lot of different nutrients. The sport is bad not to make a sport, but it's also very bad to do an excess of sport. It's quite known that people that usually they are not doing a sport and maybe at weekends they decide to do some strong sport, for example a marathon, they can develop infection, especially the respiratory system. The stress induces the cortisol, that is an immunosuppressant agent. Also tobacco, drugs and alcohol, they are all of them immunosuppressed. Very important, the vitamin D. Low levels of vitamin D has been associated with more susceptibility to develop infections and several others. In the case of age, the immunosenescence, the problem is that all people are having less naive cells due to thymic involution. The thymus is not working okay for T cells. However, we have an increase in memory cells against other pathogens, for example, cytomegalovirus. And we can have a pro-inflammation stage, and it's quite common over 65 years old to have other comorbidities like hypertension, diabetes, obesity, etc. Regarding sex, it has been shown that men are having higher mortality regarding the COVID-19. And this has been shown in different countries. In every country that has been studied, there were more male dying from COVID. This can be related with some genes that are in the chromosome X. And women, because we have double X, we can compensate if there are some, we can compensate with these genes. And also the hormones, estrogens has been shown that increase the amount of antibodies. Not just only in this infection, but in other infections, women usually get less infection diseases. This has been shown also differences in the number of receptors. Also, the tobacco habit more usual in men and other that can be more related with the sex. Regarding comorbidities, there are several elements that has been implicated, like hypertension, diabetes, hyperlipidemia, obesity, etc. And regarding genetics, the other receptor is in the chromosome X. So in the case of the case of men, it has been shown more severity, and also this protease that is helping to that the virus will go inside our cells is in the chromosome 21, and this is the reason that people with Down syndrome is having more severity of the coronavirus. Is because we are having this protease more expressed in these people, and they are more infected with more virus. And this has been shown other immune genes, for example, the type of HLA, and also some immunodeficiencies that has been associated with severity or death mediated by this virus. So the immune system should be equilibrated with moderated exercise, Food, if you are having a normal nutrition, not additional supplement is needed if the diet is correct. We should receive the vaccines, the conventional vaccines since we are children, to have a normal sleep, low stress, and avoid tobacco, alcohol, or drugs. This is one of the iconograms that we were doing during by the Spanish Society of Immunology during the pandemic, just trying to explain how to maintain a good immune system. So how our immune system works? Well, we have some soldiers that are waiting and they are very fast. They are working in the first hours, and there are several elements that are able to eat the pathogens. They are called phagocytes, and also there are other elements like complement or natural killer cells. And later on, we'll have the specific immunity, that this takes days or weeks to develop. However, there is an amplification because we have a proliferation of B cells and also proliferation of T cells. And now we can develop antibodies, specific antibodies against the pathogen, and also T cells able to recognize the peptides presented by cells that are showing like the photo of the pathogen. So the specific immune response is late, however, can induce memory. And the important thing is that we have a proliferation. So at the beginning, we have very low number of soldiers working, but at the end of the process, we end up with several cells able to recognize the same virus. So the immune response, when we have the adaptive immune response, we are having the lymphocytes, B cells, that are able, after the antigen recognitions, to develop plasma cells that they produce large amounts of antibodies, that they are able to neutralize the antigen, the virus or the pathogen, and these pathogens are going to be better internalized by other cells like macrophages. In the case of the cellular immune response, T cells are having the main role, and there is one cell that is doing the intermediate between the innate and the adaptive immune system, and it's the cell that is showing this photo of the pathogen, that is telling to the T cell the pathogen that came inside, and these T cells will develop, produce some soluble elements that are called cytokines that are going to help other cells, to B cells, neutrophils, etc. But in the case of the virus, it's important to develop these type of cells. They are called cytotoxic. These cytotoxic are able to recognize part of the virus, and they are going to release the granules that they are having inside and to destroy infected cells. So in a viral infection, we are going at the end to develop antibodies able to neutralize. We'll have T helper cell and also cytotoxic T cells. And this is what immunologists study when we want to see a specific, if this cell is specific against the virus, if this cell is specific against peptides from the virus, and if these antibodies are specific from this virus. So the immune system first should recognize that what came into it was a virus. Later on, we'll have innate immune response, and the third step is the specific immune response. So how we are able to recognize that that virus or something has entered? Well, we have in the innate cells, a variety of receptors that are not very specific. They are not able to differentiate between many different viruses or many different bacteria, but they know that it's a pathogen or is a danger signal or is an endogenous signal. So more or less they are able to recognize around 100 different patterns. And this is thanks to these 4 families of receptors that we are having either on the surface and also inside the cell. And this distribution of these receptors are mostly in the innate immune cells. And all of them start from T or C or N or R. And this is important for the recognition of the virus. However, the adaptive specific immune system, we are able to produce antibodies or T cells very specific. We are able to discriminate, for example, from the flu that we have last year and from the flu that it will be 2 years ago or 3 years ago. We can develop very specific antibodies and T cell receptors. And we can recognize millions of molecular patterns. In fact, people consider that we are able to produce thousands of millions T cell receptors on the T cells and thousands of millions B cell receptors on B cells. So after the viral entry, what happened with the cell? Well, the cell die and there is an inflammation. So the viral entry into the mucosa and also in the respiratory, so we have inflammation. But what happened with the RNA? Well, our cells are having 2 receptors, toll-like receptor 7 and RIG1, that is able to recognize this RNA. And what happened? When a cell recognizes that this is a virus, try to call to the neighborhood, to the other cells around, releasing interferons, and is telling to the other cells, be careful, inhibit your protein synthesis because there is a virus around. So try to call to the neighbors to avoid the infections. So interferons type 1s are very, very important to stop at very early age the further expansion of the virus. And this is the reason that when you have genetic defects affecting this toll-like receptor 7, the RIG1, or the production of interferons, for example, because there are people that develop autoantibodies. We develop antibodies against our own interferons, and this has been shown that it could be even in 6% of severe cases in older than 65 years old. So these autoantibodies are very, very common. These are related with severity or death. So interferons 1 are very important for the antiviral response. Here is just showing how the cell, recognizing the RNA, produce at the end the production of interferon type 1. And this interferon, what the effect is that this cell is already infected, but try to call to the other cells around to do the inhibition of the viral replication. So timing is very important, and it was shown that interferon 1 should be produced very early on, because if we have a delay in the signaling, we can have inflammatory and a dysregulated immune response. So it should be produced at very early stages. The second stage is the innate immune system. We have this macrophage, neutrophil, monocytes. These soldiers are able to phagocyte or to kill infected cells. And also we have some elements like complement and cytokines. The macrophages are able to produce soluble substance. And if they produce a lot of cytokines, they produce pro-inflammation. And this is a really big problem in the lung because it can produce what is called the cytokine storm. And what happened in the patients? Well, the number of monocytes in patients that are critical are low, but increase some cells and also the production of interleukin-6, one of the pro-inflammatory cytokines. And this has been correlated in moderated and severe COVID with the inflammation that is produced especially in the lungs, with high levels of cytokine, and is called cytokine storm. So this is associated with high levels of interleukin-6 and also other cytokines like TNF and increase of C-reactive protein, etc. And people that die is having this severe COVID because of this problem. Regarding natural killer cells, natural killer are cells that are able to kill. At least we are telling all the time, please don't do it, don't do it. We have to stop them, and we stop showing on the surface some elements and stopping giving negative signals. However, if a target cell, like for example infected or tumoral cells, is not providing this negative, this natural killer release the granules and kill the target cell. Well, but the natural killers are very important. However, it has been shown that there is a decreased number of natural killer cells, and also they are exhausted. This can be shown by several markers on the surface, and there are several papers that has described this decrease in the number of NK cells. And this decrease is correlated with the severity of the illness. So natural killer seems to have a very important role in the protection against the infection. The complement is a mixture of several proteins, serum proteins, that they can be activated by several elements, directly by microbes, by bacteria, also by antibodies recognizing some pathogens, and also sugars like mannose binding to lectin. And at the end of this process, we are going to have the release of some factors that are like bells calling for other cells. They are mediated inflammation. And also there is opening of holes, pores, on the surface of the microbes. So the complement is very important to kill bacteria or virus, and also as a bell to call cells coming to the inflammation. Well, the complement has been also correlated with the process of the inflammation in the lung, and there are some papers that have correlated the high levels of C3A, 4A, 5A with inflammation in the lungs. So in general, as a summary of this part, the severe patients are having an affected immune system with a pro-inflammatory state. There is a decrease in the number of T, B, and NK cells, high inflammation, and this is related with the immunopathology of COVID, with this cytokine storm, with high levels of cytokines, increased number of antibodies, and also the monocytes and the neutrophils has been implicated in the thrombus formation because an inflammation in the endothelium is able to activate tissue factors and started the coagulation pathway by the extrinsic and also by the intrinsic coagulation cascade. So all these elements are participated either in the response to the virus, but also in the inflammation. And later on is the specific immune response. As I said before, they start working later. We have to wait several days, weeks, but at the end we will develop antibodies of different isotypes. The first one is M, then we have IgG and IgA, and we can develop T helper, cytotoxic, and regulatory, and also a specific memory T and B cells. But what happened with severe patients? That there is a decrease in the number of cells, that you can see this in these red dots. There is a decrease in B cells, in gamma delta, in cytotoxic helper. In all the cases, there is a leukopenia. The virus is affecting the immune system. And severe patients are higher levels of antibodies. They are, because they need to respond, and they are having virus, they are producing virus, they are producing antibodies trying to neutralize the virus. However, people asymptomatic, the level of antibodies are quite low. This is another journal showing that if you can see that in orange is people without symptoms, asymptomatic, they have very few antibodies. However, in the case of the people with symptoms, they are having much higher levels of antibodies, either IgM, IgG, or IgA. And what happened with other coronavirus? Well, with other coronavirus, the natural infection, we know that the antibodies usually stay for 6 to 12 months. And in some experiments that were done in 1985, infected volunteers with some coronavirus producing a common cold, they were showing that the levels also decrease around 12 weeks, and then there are low levels at 6 months. So it seems that the normal infection, but other coronavirus, they don't stay, at least in the antibodies, very long. In the case of COVID, it has been shown that there are production of antibodies and immunological memory at least until 8 months after infection, and this was confirmed by several studies, including a recent Spanish study. So what we are expecting from the antibodies? Well, the antibodies can neutralize directly, they can attach to the virus, and this is something that we want because we avoid that the virus will enter inside our cells. We want to form the immunocomplexes and that the phagocytosis will be efficient, removing this virus. And also we can have the activation of the complement cascades that will help to destroy the virus. And also the antibodies help to natural killer cells to destroy the cells infected. The antibodies are making the bridge between the NK cells together with the infected cells. And these NK cells will release the granules to kill the target infected cell. However, not all antibodies neutralize the virus. We can have several antibodies. In fact, most of our antibodies, they do not neutralize. There are some of them, especially those that avoid the interaction with our receptor. But we should take into account that it's not the amount of antibodies the most important. The most important is to analyze the neutralizing antibodies. During this pandemic, some people were saying, be careful, because to have a lot of antibodies maybe could be even dangerous, because it has been shown in some viral infection an effect called antibody-dependent enhancement effect. And this is related that there are cells that are having receptors for antibodies, And these cells can become more infected and with a more pro-inflammatory profile, because we are continuously infecting these cells and they are producing cytokines. Another situation is that antibodies, soluble antibodies, can interact with receptors on the surface of B cells, giving negative signal. So soluble antibodies are telling to this cell to not become activated because we are already here. This is what is called antigenic sin, and this is very important when you want to develop a vaccine. So the Fc receptor function is very important also in the regulation of the antibody response, in the phagocytosis, in the maturation and activation of the dendritic cells, and also in the granulation of eosinophils and neutrophils. So these receptors are quite important. And so what we know about people infected in the COVID-19? Well, we know that they develop T helper cells in 100% of patients having infection. In this paper that was published in Cell last year, 100% were having CD4 cells, helper cells, recognizes not only the S proteins, but also other proteins from the virus. However, the antibodies were able to recognize this S protein. They were also able to recognize cytotoxic T cells, either S antigens, M, N, etc. So even these people, they claim that vaccine should include other antigens, just not only the S protein, because we are able to produce cells able to recognize other antigens. In the same paper, they were showing that could be some cross-reaction, and people that were infected by common cold coronavirus, they were having also some cells, CD4 and CD8 cells, able to recognize also the SARS-CoV-2. And they were thinking that this was due to cross-reaction. So the whole immune response is working something like that. We have infection, the dendritic cells internalize and they migrate through the lymphatic, to the lymph nodes close to the place where these dendritic cells were activated. There is a presentation to the T helper cell that is able to help to the cytotoxic cell to become completely activated, and they can go to the tissue to kill the infected cells. However, they can collaborate with the B cells, and these B cells become plasma cells able to produce antibodies that they will neutralize the virus. So this is how our immune system works. And this is how it works when we use a vaccine. What we want to develop memory against a pathogen. And there are two types of memory. One is the classical one, and more recently it has been recognized one that is called trained immunity. But basically we develop memory by two ways. Having the infection, passing the infection will be the natural way, or by vaccination, using attenuated death pathogens, subunits, can be proteins, toxin, DNA, RNA, peptides, whatever, but they will not induce the illness. Could be some secondary effects, as all of us we have with the injections, but we will not develop the COVID, the severe COVID. And the memory T and B cells reach the highest level around 1 month, and they can be maintained for long periods of time. How long? This is what we don't know, because we don't have enough time to know how long we have. In the case of the trained immunity, it has been described for natural killer cells and for macrophages. And people have thought that maybe it could have an important role in the COVID in children because they are not infected very much or very minor. Also, they were saying that because children, they were receiving recent vaccine, or they were having other infection by coronavirus, or they were receiving the BCG vaccine. Well, it's not completely clear, but it's true that children are having an innate immune system more potent than all people. So it could be due to this trained immunity. But how classical memory is maintained is not completely clear. Some people think that we develop just from the very beginning long life memory cells, that they can live more or less forever, and developing long life plasma cells. Other people consider that it should be an internal persistence of low level of antigen. So you have a little bit internally, okay? So our infected cells, they produce a little bit of virus, for example, and at the end we have this the maintenance of memory. Other people consider there is a cross-activation by other antigens. So these cells, the memory cells, are more promiscuous. And even if appears a virus that is different or other pathogen, a bacteria or whatever, is able to activate. And something that it has been shown that is not necessary is that repeated exposition to the same pathogen. For example, yes, if you have an infection by the flu, to have against the same flu and against the same flu to maintain this memory. But something that is important to know is that to develop memory B cells, we need to have the germinal center development, that is a structure that you can develop in the secondary lymph nodes or secondary lymph organs, including spleen, lymph nodes, player patches, etc. And you have isotype changes. You can incorporate mutations. These cells can mutate in the immunoglobulins, and they have a lower requirements for activation. So this is what is a memory cell, able to have on the surface a different immunoglobulin and able to have mutations. And this can derive to plasmablasts and give place to long-lived plasma cells. If they go to bone marrow, if they go to mucosas, or if they go to spleen, they are able to recognize different isotypes. In the case of T cells, they are more difficult to study because they don't change the class and they don't mutate. So the only way is to check for some markers that are known, that are expressed if the cell is a stem, or if it's central memory, or effector memory, or resident memory. And there are some markers that has been described that can distinguish different subpopulations. So what we know, and now I would like to compare what happened with the natural infection compared with the vaccination. Well, the natural infection, we know that gives a very variable immune response. There are people that has severe leukopenia, several factors that affect, so there is very variable, the immune response. In some cases, it's very, very minor, in children, for example, and in all people are having a very, very deep effect. How long is the memory for the natural infection? We don't know. And we could expect that it will be variable because there are people that maybe have been more affected than others, and maybe some of them have mobilized more memory cells than others. Regarding the variants, reinfection is possible but rare, and the variability also it could come from new variants. For example, if one person were infected with the alpha and now comes a new variant, it could have reinfection depending on the variant. It's important to say that the natural infection induce a more broad immune response in the sense of epitopes, in the sense of antigens that are recognized. Because T and B cells are against different viral proteins, as I was showing before. So we have T cells against S and M and antibodies against the spicula protein. What we know? Well, but at least 8 months, this immune memory we can recognize memory cells, memory CD4, memory B cells, and plasma cell-derived IgG antibody. And it's different if you have mild or severe, and they have this paper have shown that there are large variety, and especially severe COVID, there are people develop immune response against different proteins in the virus. We don't know the long-term memory. We don't know what will happen if these memory cells will stay for long. This is something that we don't know. What we try with the vaccine, especially with the messenger RNA vaccine, what we want is that the dendritic cells will capture the antigen, will migrate to the lymphatic, to the lymph node. They will present and develop the germinal center reaction. So at the end, we will have plasma cells developing antibodies, and also we'll have CD4 and CD8 activation, producing cytokines and killing infected cells. So the image that I saw before of the immune response is the same as that we are expecting with the vaccine. What happened in the immune response against vaccine? Well, we know that gives protection against severe illness, decreased secondary effects and deaths, and the response is less variable. Why? Because we are injecting the same amount in every person. We have a potent protective immune response, and also increases this immune response in persons previously exposed to the virus. How long? We don't know. We have been given 2 doses of Pfizer, Moderna, AstraZeneca, or 1 dose in the case of Johnson & Johnson. We know that protect during several months, but we don't know how long this vaccine will protect. Variants. Reinfection is possible. It has been shown in aging and immunosuppressed people with some lower responses against some variants, especially the Delta. And this is telling us that vaccines maybe should be modified against new variants maybe in the near future. And something that also is important is that the vaccines, all of them at the moment, are containing just only the S protein. So the antibodies and the T helper and T cytotoxic cells are only against this S protein. How long has induced the memory? Well, there are several papers. This was very early on, the 10th of February, in very few people, but they were saying that they were neutralizing antibodies and proportion of memory B cells were similar in vaccinated people to infected patients. In this paper, they were saying that there were better responses in vaccinated than in infected patients. There are some controversy about that, but what is clear is that the vaccination induce something that we see in the serum, that are the antibodies, and these antibodies decline with the time, that's for sure, but there are many things that we are not seeing, that is like in the iceberg, is in the shadow. At least in the immunology laboratories will study what happens with this memory, B and memory T cells. One study that Rita Carcetti has done is that people that were previously infected and they received the vaccine, they increased the level of IgA in saliva. And vaccines, as you can see here, in the level of IgA that is produced in the mucosa, they are very low amount of IgA, but if people are previously infected after vaccination, the levels of IgA increases in saliva. So this is telling that infection alone can receive a vaccination after that, and the persons could be more protected. Regarding vaccine efficacy, I don't know if you have heard that Israel was saying that they were having problems, that the vaccines were losing efficacy, and that it was required a third immunization. Well, a very recent paper on 13th of September in Lancet, they were testing against the different alpha, beta, gamma, and delta, and they were showing that the vaccines were very efficient to prevent severe disease. And they were testing, as you can see here, different vaccines carrying vector, inactivated protein subunits on messenger RNA. All of them were very effective to prevent severe disease. And also, it was by statistic in United States that was saying that the analysis in Israel were not very well studied. And using the same data, but doing a stratification in ages, and with the percentage of population that were non-vaccination or fully vaccinated, they were showing that the efficacy of the vaccine was over 90% in most of the cases, and they were just only a little bit of decrease in people over 80 years old. So this is telling us that we should be very, very careful with how the data are presented, and the alarm that this mobilized several countries against the use of a third immunization.
Africa, sorry, we are approaching our... Okay,
so I have to finish, no? I should finish. Okay, well, yes, I think I have just related with reinfections, that there are very few reinfections after passing the COVID, and after the vaccination offers large protections. And that is quite recent, but it has not been reviewed. Against the variante Delta, they say the natural immunity gives better protection and duration against the Delta variant compared with the Pfizer vaccine. And well, this is the result that vaccinated people infected are having higher levels, and receiving the vaccine are improving and induce proliferation and more antibodies than naive subjects. And well, we should know that this is not the last pandemic, and that maybe in the near future we could have different ones, from coronavirus or from other flu virus. So sorry, because it was it took me more time than I expected. Thank you very much. And if you have questions, I will be happy to answer.
Thanks a lot, Africa, for your very comprehensive talk on COVID. the subject that is, I'm sure, conditioning the lives of everybody here, and we are now much more well informed on how the immune power deals with the disease. Okay, we have a question from Alexandra Moreira. Are you in favor of a third vaccine dose?
I don't think that it should be for the general public. I think now we are in the moment that maybe we can study several situations. Of course, for immunocompromised people and people with immunosenescence, the third immunization is just to complete the vaccination. But I don't think that the third dose should be generalized. At the moment, we don't have data that is telling us that the vaccines are losing efficacy.
We have another question from Lydia Gonçalves. Do you think that the IgA at the level of mucosa will be important for virus neutralization, so that vaccine route or immunization will be important?
Of course. IgA, if we have IgA in our mucosa, is able to neutralize just immediately when it's recognized the virus, and you should not have the infection because you neutralize immediately. The problem is that intranasal or other routes different to the intramuscular are having problems. The intranasal administration, there are just only one vaccine for flu in the United States that in Europe has not been authorized. And there are problems because the nose is connected with the olfactory nerve, and it has been shown that can go to the brain. So that's the reason that the regulatory agencies are very aware, they are scared about the intranasal administration. Regarding mucosal or inhalatory, this could be as well another situation, but we should have this type of vaccines, and we don't have very much expertise. And we know quite well that the intramuscular is giving IgG, is able to have systemic, some IgA, not very much. But I agree with you that the important is to have IgA at the level of mucosa.
Another question from Begoña Pérez Cabezas. Why not analyzing the cell immune response instead of the antibody response to understand the duration of the response, so the lower part of the iceberg?
Sorry, can you say again why you don't analyze the cellular immune response?
Probably the question is, why are we that much concerned with antibodies and not with the cellular response as well?
Well, to study the cells are more difficult. It needs more expertise to do it in laboratory, and not all immunology labs are working with cells. Now they are trying to develop some alternative ways. For example, there are cutaneous, like Mantoux test. One Spanish person, Ibelice Barrios, has developed a cutaneous test to analyze the memory in response to several antigens. And also they are doing like, how it's called, a quantiferon test. They are doing something similar. So you can activate the cells with antigens, and then you check in ELISA how much interferon gamma the cells are producing. So now they are trying to find alternatives that could be easier. But at the end, you need expertise and a special laboratory to have the whole picture of how our memory cells are.
Another talk. Do you think more should be done from South Korea? Do you think more should be done to fight misinformation about infection in vaccinated people?
If we should give more information to the people, no?
Actually fight misinformation.
Okay. Yes, fake news, no? To avoid fake news and all these kind of things. Well, I have been trying during the pandemic to talk with the media, with the journalists, and also in several media, writing, mass media, television, etc., trying to resolve doubts. I think that we should teach more about immunology in general. The immune system is forbidden. We don't know that we have. We know that we have liver, that we have heart, but we don't know how our immune system works. It's terrible, and we should know more about our body. And the immune system is in the base of many illnesses, and we should know more about that. And I hate many of the news about this food or this nutrition is is good for your defense. No, it's now everywhere. This is very good for your defense, and nobody has tested if activate the immune system or is having really a role. I think we need to educate more people, just to the children, to go to the schools, to the high school. Immunology should be more present in our lives. And maybe with this pandemic, people will start being more conscious that the immune system is important, how we defend against the infections, how the vaccines are made, all the controls that are required. I don't know, I think this is an important moment for science in general and for immunology in particular.
Africa, do you think that people is now more informed about what immunology is about? Because even the title of your book is Immunopower, so we have a tendency to look at the immune system as something that fights whatever attacks us. But as you clearly show in your talk, the immune system is also about regulation. It's about harnessing this big army that is very successful in fighting pathogens. Can you comment on that? Do you think that people is as well aware that we have to regulate, not only boost immunity?
Well, I think that people do not know that. However, for example, showing the cytokine storm, no, saying, you can have a crazy immune system. You can call it like that, no? Or when you have autoimmunity, you have also crazy immune system. It's true that we don't talk about much about regulation, and in fact, just not to talk about the book, but I decide to include one chapter. The whole chapter is related with regulation because, as you said, is very important. It's very important how to activate the immune system, but also it's very important how you switch off and stop the activation. Otherwise, because you have the T and B cells that they proliferate, you can have a positive feedback that can be very, very dangerous, very dangerous. So I completely agree with you that we should teach more that is homeostatic. It's a homeostatic system, and the pathogen is something exogenous. The immune system is having a role, an internal role, a homeostatic role, and the pathogen is altering this homeostatic, But this is not like we have our immune system just waiting for the pathogen to come into. No. We have our immune system for our the control of the organism, that everything will go okay. It's cleaning all the dead cells, are checking that everything is okay, and it has a very, very important role in regulation. Yes.
We have another question that is, I guess, a very important one because it comes from our students from ICBAS Amphitheaters. And the question is, what is your opinion in relation to vaccinating children?
Okay, it's a very good point. I am not in favor of the immunization of children under 12 years old for several reasons. There were several papers checking that the children they are not so contagious as adults. They don't have many virus in their mouth. And also they are having very mild infections. In fact, most of them are asymptomatic. At least with the variants that we are having now. If new variant will come and the situation will change, I will change my idea. But at the moment, what we know is that vaccines are not stopping the infections. One person that has been vaccinated can be infected and can spread the virus to other people. However, the vaccine is decreasing the severity of the illness. And if a child is not suffering from illness, why to use a vaccine that is doing that effect? And also, we should be aware that vaccines can have secondary effects. It has been described some myocarditis, inflammation but that is not in a very high number. It's quite rare, but it's there. There are some secondary effects that we should, and also we don't know. They have not been accepted. The European agency, because we don't have the data of under 12, but the number of children that has been included in the clinical assays are very low. So we don't know exactly the security. We have very low number, and maybe the secondary effects could be different to adults. We don't know. So my view is that under 12, no. And why over 12, yes? Because over 12, they are more like adults. They have more virus. They can be more contagious. It was a paper putting more or less around 10 years old. 10 years old is under 10 years old. They are less contagious than over 10 years old. I think 12 years. It's a time that is nearly to the adults, and they can have also the infections can be. There are some cases of severe. So I think that over 12, I agree with the vaccination in children older than 12.
And another question, what about pregnancy?
Okay, pregnant woman has been shown that they can develop a very, very severe COVID, very severe. And there are some pregnant women that have died with COVID. So this was an emergency saying, look, we are seeing that there are many pregnant women dying, so these women should be vaccinated. And this has been shown that are protecting the fetus, and also there are antibodies in the milk. So the mother, when is breastfeeding the child, is protecting with IgA passing to the child.
We have another question from Olga. Could there be deleterious effects in less severely affected patients?
Can you say again?
Could there be deleterious effects in less severely affected patients? I'm not sure if it's the disease or vaccination that is at stake here.
The question is if we can develop less deleterious
if
vaccine.
People with less severely infections can develop deleterious effects, probably that.
I don't know if they are saying that a person has become infected and then received the vaccine, if they are having more secondary effects. Is that the question?
I'm not sure if it's the vaccine or the disease.
Or the disease. Okay, well, what it has been shown is that people that were having the infection and then they received the vaccine, they have a very high response. So that was the reason that they decide to delay at least 6 months from after the infection to wait 6 months to receive the vaccine. And if they are under 65 years old, just to receive one shot. And if you are over 65, to receive 2. And it has been shown more secondary effects especially in young people, in those that have a very good immune response. There are more secondary effects in young than in older. I don't know if I ask your the question.
Yeah, I guess so. Okay. Oh, so it's sequelae actually, the point here. Even people that has less severe illness could afterwards develop side effects, sequelae of the disease, even if you have a milder disease.
There are people that were having very few symptoms, but these symptoms are maintained. To be honest, it's not well known how the long COVID, if it's due to the immune response, if it's due to the permanence of the virus in some areas in the body, because it's known that the virus can stay in our intestine for quite a long time. So I think that now is the time to study the long COVID. I don't know. I don't know if what would happen in the future. I have seen some papers saying that if these people that were having no very much symptoms and they were receiving the vaccine, the symptoms were decreasing. In some cases, the vaccines were like removing the virus, no, and they were feeling better. But I'm not sure that this has been done with many people. I cannot be sure that this will happen in everybody.
Okay, we have another question that must be the last one because we have really to finish. Comes again from Alexandra Correa. Do you think we need new vaccines or the improvement of current vaccines?
New vaccines or improvement of the current ones? Very good question. I said last year when people were asking me about vaccines, I was telling them the first vaccines will not be the best, but they will be like we have, and we have to use them. Of course, maybe they are not the best. And I was just saying during the talk that for me, maybe the best vaccine is something that will be more similar to the whole virus. Just not only having the S protein, that should have more things. And of course, going to the S protein was the faster way, and this has been amazing. Scientifically talking, it's absolutely amazing that, and thanks to Dr. Carico, I'm very bad that she has not received the Nobel Prize this year, but thanks to Dr. Carico, that she has been working for 30 years, that we have the RNA vaccines in less than 1 year. This is absolutely amazing. But this is not, maybe this is not the best vaccine, especially for maybe for long period of time. We don't know. But I can tell you that a vaccine that will give a protection over 95%, like Pfizer, is very difficult. Very difficult. And many people that are trying to work with other vaccines, what they are having is 80%, 70%. To have 95%, like the Pfizer, and Moderna was 93%, it's very, very difficult. New vaccines maybe could give more longer protection along the time. But as these vaccines, they are really, really very good. So I don't know. We should wait to see the results with other vaccines. Now, to do the studies are more difficult. They have to compare with already vaccinated people or going to countries where they have not have people vaccinated. And you don't have virus moving around very much. So the studies are much more difficult.
Okay. So we have another last question that comes from Coralia Vicente, former director of the ICBAS, and she asks, what would you advise us as a school to do to pass the correct information to the youngsters and to the press? What can we do as a school at ICBAS to contribute for good information?
I think we can contribute with for example, with this webinar, with allowing people ask questions. Sometimes they think that they are not good questions and they don't want to ask. Just to have children around that they will ask you whatever they want. And I think in close contact is better than in maybe in journals. I have seen that, for example, the anti-vaccine groups, sometimes when you are sitting with them and you show the results, you dedicate time and you explain things, sometimes you convince them that to have the vaccine is a good thing, you know. So I think to go to the schools, to sit with them, to allow them to ask questions and to in a very informal way, maybe is good. And to avoid all the bulos, I don't know in Portuguese or in English, bulos and fake news and all these things. In Spain, it was a program that every day is saying, this is a fake news, this is fake, this is fake. So trying to stop it as early as possible. Hypochlorite is not killing the virus, for example. Just telling very, very quickly, just stopping the stupid things and many things that they were going around. So I don't know, but I think going to the schools, going to the high school maybe is the best. I think that people seeing the television, I don't know in Portugal, but in Spain, it has been routine every day, talking to the experts. And I said, there is no expert in a pandemic like this. None is an expert. We are learning every day. We don't have any expertise about this virus. So, I don't know, it's something that we should do as immunologists. I think we should help to people to understand and at least to understand few concepts that are important. And being honest, we don't know how long the memory will be. We don't know if this vaccine is the best. We don't know if a new variant will come. Saying the truth, I think, is the most important.
But journalists will not be happy with this answer, right? We know that. Okay, so thanks a lot, Africa, for your kind participation.
Thank you for your invitation.
You proved that it was the right person to start this webinar series that will go on. Next invitee will be Professor Nicholas Proudfoot from University of Oxford, and the session will be chaired by Alexandra Moreira, that is putting forward these webinars. So at the very beginning of your talk, we had a message from Washington, D.C., an ICBAS alumni that congratulated us for the opportunity to reconnect with the school. So the webinar has been a success since the very beginning. And this is also good to put people in contact.
Thank you. Thank you very much for the invitation. It's a pleasure, as you know. My English was a little bit rusty because with the pandemic, I was not traveling a lot. So I hope that you understood everything, because sometimes it takes time to adapt the brain.
Portuguese adapt very quickly to this language and accents. Questions. So it was perfect, African. I have to say that the feedback that we received and that most of us saw in the chat is very, very positive. So it was really a great start for this webinar series. So thank you very much, Africans. Thank you very much to all those that attended this session. I hope that we could count on you on the next webinars. And okay.
Okay.
Thank you all, and bye-bye.
Thank you. Bye-bye.